Good evening and welcome to the third in our Frontiers in Chemistry webinars. It's wonderful to see so many alumni and friends joining us this evening for what promises to be two very fascinating talks. These webinars are made possible by generous sponsorship from Dr. Yusuf Hamid. He has sponsored this outreach event since 2021. And as you know, he sponsors many other events in the department. We're very grateful for him for his continuing support. Now, tonight's talks are really about color. And color is something that chemists have always been very fascinated by. What is it about one structure that makes a substance red and another blue and another green? And how can we manipulate these structures to create different colors? Now, of course, in the 19th century, this idea led to the development of the first synthetic dyes. And I hope to show you one here. This is Movine, which was synthesized from coal tar by Perkin in 1856. And this led to a complete revolution in the available colors and dyes. And of course, the development of a very important chemical industry, which has continued to this day. But of course, the problem with these colors and pigments is that they are not really sustainable. They're not made from sustainable materials, and they're certainly not biocompatible. And that really brings us to the topic of this evening's talks, which are addressing this issue of how do we create color in a sustainable and biocompatible way. And in this department, Professor Silvia Vignolini and her group have been studying this problem. And what we're going to hear about is the solutions that they have come up with. Because by drawing inspiration from nature, they've realized that you can create color in ways other than using dyes and pigments. And I will leave Silvia and her team to tell you about how that is. Because they are inspired by nature and made from natural materials, they are, of course, inherently sustainable and biocompatible. So we have two short talks this evening. The first from Professor Vignolini herself, and that's entitled, as you can see now, Making Colors with Your 5 a Day. And then after that, we will have a following on talk from a senior postdoctoral worker in her group, and that's Dr. Thomas Parton. And he will talk on self-assembly with a twist, which is about making photonic pigments from cellulose nanocrystals. I'm sure you'll find these both very interesting talks. After the talk, there will be a Q&A session, which will cover the topics of both presentations. If you wish to make a comment or ask a questions, please type it using the Q&A box that you will see appears at the bottom of the screen. And finally, just to remind you that these presentations are being recorded and they will be shared online following this event. So without further ado, I will hand over to Silvia. Please look forward to hearing your talk, Silvia.
Thank you very much for giving me the opportunity to talk at this alumni event. And I just want to give you a little bit of a brief introduction of what we do in my lab and why we study color. So we really look at how living organisms that we find in nature, that goes from plants to insects to even bacteria, manipulate material at the scale of a few hundreds of nanometers to interact with light in order to produce really interesting and different type of optical appearance. And as James just said, the reason why we try to do that is because we want to produce material using a building block that we borrow from nature in order to make color in a more sustainable way. And I really became fascinated by this topic when I started to look more at how different organisms really manipulate their appearance. Now you will see in a moment from this video that what you think it's a dull color from an algae is actually the coloration of this squid that is able to camouflaging, changing completely the texture and the appearance of this coloration in real time, really fast, and actually using only a really simple system of protein and a combination both of pigments, but also of what's so-called photonic structure that I will show you in a moment. So why we are interested about color? Color it's a really important form for communication, not only for animals, like in the case of the squid, but it's really important form of communication for us. If you simply look at this picture of a supermarket where many different types of color and combination of color are used to attract our attention in packaging, you immediately understand that the reason that it's important to be able to make color more sustainably. It's really a topic that we need to address. We need to address now. So maybe I will just go back one moment to show you this video, because I think it's really worth seeing, because it's something that we are not often used to seeing, the type of color that we have available today. Yes, material that are capable to change their optical appearance in such a fast response, and material that are made out of building blocks that are fully natural actually are really uncommon. And as I said, so far we are used to think about color in terms of pigment, and this is also true for many of the color that we see around us, especially also on the things that we eat. But actually, it's also possible to make color in a slightly different way and using material that are fully transparent. This might sound a bit strange or not really familiar to you, but when you look at a soap bubble, essentially, this is actually what is happening every day in our kitchen. The liquid of water and soap, it's by itself transparent. If you have it in a glass, you don't see any color, so it's not a chemical characteristic of the material. But as soon as you make a bubble, you are creating a thin film. And the length scale of this thin film is of the order of magnitude of the wavelength of the light. And therefore, you can interact with light with a mechanism that is called the interference. And you can actually create coloration out of material that by itself are transparent. So what is the message here? What is important to remember is that actually you can make color with any type of material, even transparent material. And this is a strategy that is extremely exploited in nature. In this slide, I want to give you some of the example of the variety of the material and the species that use this concept of photonic structure in order to make color. So as an example, in many weevil, when you look at them closely, you look at this small scale, but also similar in butterflies. You have photonic structure that are made of a combination of chitin and proteins that on the nanoscale can produce this really strong, brilliant color. Similar, maybe you are more familiar with what you observe in birds. Many different birds, including common pigeon, they have some feathers that are really strongly colored and tense. And this strong coloration come again from this special interaction with the light and the material. And I would like to focus your attention on the birds, because this was first discovery of structural color that was made by humankind. And this is almost 2000 years ago. In fact, if you read the philosopher Lucrezio, who wrote the De Rerum Natura, in one of the books, he started to describe the color of the peacock, and he was really fascinated by this color, because already back then, he actually realized that was something strange going on with this type of coloration, because in contrast to any other coloration that he had experienced, this colors was changing a bit with the angle of observation of the observer. And I think you are also familiar with this type of experience, but for me, it's quite fascinating that already 2000 years ago, someone was able to then not to explain the physics and the concept behind this observation, but yet saw that the structural color are somehow unique and have unique feature. And this is actually one of the features of the structural color, that you can get really strongly bright coloration, and also you can also obtain this effect of color change in function of the angle that is called iridescence. Interestingly, if you look also in mammals, so not really far from the phylogenetic tree from us, they can make structural color, and they can make it with a material that is also we have available in our skin, that is collagen. So this blue coloration that you see near the nose of this baboon, essentially it's obtained by a collagen structure, again, on the size of the wavelength of the light, that interacting with light produce this blue coloration. And finally, what I think it's more interesting for what we are discussing this evening and what we do in my group we look a lot at the structural color that are produced by plant. And we focus on plant because at the end, it's a material derived by plants are generally very, very available. And we have started this work actually in strong collaboration with the Botanic Garden in Cambridge. And if you have the occasion, I strongly suggest in the summer to visit, because some of the fruits and the material that we got inspiration and that we are also studying in details are essentially coming from the Botanic Garden. And what is interesting is that all this blue color that you observe in this slide that are coming either from fruits or from special leaves are not obtained by any pigmentation, so by a molecule. They are coming from the specific type of structure of the cellulose organized in the cell wall. So you might know that cellulose is it's a really abundant material that is present in any plant cell wall. And it comes out and is produced in fibers. I will show you more in detail later how does it look like. But what is important is that in this specific type of fruits and plants, these fibers are organized in an architecture that we call helicoidal. Why it's called helicoidal? It's called helicoidal because it looks like one of these staircases that are somehow stacking on top of each other at a slightly different angle, forming a kind of an helic structure. And when this helic is comparable with the wavelength of the light, so the size it's more or less the same, you have you can produce an optical appearance and you can produce discoloration even in absence of any pigment and with a material that is cellulose that essentially it's by itself transparent. And we started to look at the optical properties of these fruits when I started my group here in Cambridge, and we got really fascinated on how the different mechanisms interact, how it's possible to build up in the cell wall. And then from this, we started to ask questions, can we make color in the same way that nature can do? Before I tell you a little bit what we can do, I want to tell you also why this is really important. And as James just flagged briefly before, the way we make color today, it's really unsustainable, especially in the case of this metallized color that we often see in different pigment cosmetic products, but we also have in many of our cars. And these one are generally obtained in two ways. One, it's really that is also similar to plastic glitter, using multi-layered structured metallized plastic film that are doped with dyes. And these have a double problem. The first, that they contribute to the pollution of the microplastic pollution. And the second problem is also that you have to manufacture dyes, and the manufacturing of dyes in general, it's an industry that it's really difficult to integrate with the principle of green chemistry. So it's not a problem that has been completely solved. The other way that people use to develop this metallized pigment that we commonly have in our car on these products is to use a different layer again of mica or titanium dioxide and different inorganic material deposited on top. And this is again problematic, especially for not only for the environmental cost, because you need to use it at high temperature, but also the material that are used is inorganic materials. Some of them, like titania, have been recently banned by the European Union because they are not all problematic for their environment, but also they have been demonstrated to be carcinogenic. So this is like a problem that is not 100% solved. The idea that we had is like, can we make color in a more sustainable way? Can we like somehow try to do like nature does? Because if you look at so as I was telling you before, the majority of the synthetic pigment today, obviously some of them are not toxic, but lots of them, they are produced with toxic chemicals. And despite some people have proposed plant-based or microbe-based structure to extract natural pigment, in terms of sustainability, this is not really like a solution that can be applied on large scale on the market. Mainly because you need, for example, if you do plant-based one, you need to have soil and reserves area to cultivate these plants to extract the pigment, and the same soil and water can be used to actually cultivate food that is used in the growing for our society to feed the growing number of people. So the idea is that if we could really reach this sweet spot where we can use non-toxic material, but sustainable material that are really available, we could solve a problem that it's really actual. So, and our solution that we thought that it's a really good material to use is actually cellulose. And why it's cellulose? Cellulose is the most abundant biopolymer that we have available on the planet. In fact, it's present in any plant cell wall. And when you say that you have a diet that is rich of fiber, these fiber are essentially cellulose fiber. And this is the reason why cellulose is not only a biocompatible polymer, but it's actually also edible. And cellulose, because it has been exploited by humanity in a huge variety of contexts and in many different ways, is one of the most exploited material also for society, from textile. You can think about all the cotton-based textile that we use, to like my glasses are made out of cellulose acetate. That is one of the cellulose derivatives. So cellulose itself, it's a material that means a lot of different materials. In the plant itself, cellulose can be found in a completely big variety of different forms. You generally have fibrillar structure, but they can organize in different architecture. One I showed you before that can produce color. The fiber can be larger or smaller. It really depends on the source. But the main things is that you have essentially is produced by at the cell wall membrane by a protein complex, and it's pinned inside the cell wall as a sort of a hierarchical fiber itself, made of polymer chain that are essentially glucose molecules linked together. So there are different types of cellulose, and within the different type of cellulose that we use tonight, I want to just show you a couple of them that I think are particularly interesting. I want to start with the hydroxypropyl cellulose because, and you see HPC, and many of you are chemists, so they know better than me that this HPC is one of the most used food additive, and also in the pharmaceutical industry, one of the most exploited material. In fact, you might have eaten the hydroxypropyl cellulose if you had a tablet of aspirin, or you might have eaten sometimes used to thickening cream or ice cream, and also if you are gluten free, the main ingredient of gluten free pizza is actually hydroxypropyl cellulose. Yeah. So it's a material that we know that is edible, but maybe what you don't know is that if you mix this hydroxypropyl cellulose simply with water, you let it rest, what's happening, you start to form this beautiful coloration, and this they appear just letting the suspension rest for a couple of days, high concentration, 60% hydroxypropyl cellulose and the rest water in a vial. So what's happening? If you look macroscopically at this film, and you look at how the polymer chain are organized in this type of system, what you notice that they are actually forming an helicoidal-like of architecture that in this case you would call it liquid crystalline phase in chiral nematic phase. And what is nice is that you can change the dimensionality of this phase by changing the level of water present in your film and the hydration. And you can see here that you can tune the range of the color from the infrared to the ultraviolet light by simply change the amount of water that you put. And this color also is particularly special. So we said that we change in presence of water. If you add water, the color becomes a red shift. So you are increasing what we call this chiral nematic structure. But then if you apply, for example, a compression, locally you can somehow squeeze the molecule out of this architecture, maintaining it, and you can change the color. And this is just a really nice small demonstrator that we built that you can see when you press it, the color change. And then later that we really try to exploit this as a real material for sensing application. And we were able to make it on a really large scale, maintaining what I think is the most important part, that actually the material, it's edible. And these are an example of different cakes and chocolate decoration that we have done at the Christmas party and with the group. Another system that we used a lot is they are called cellulose nanocrystal. And the cellulose nanocrystal are essentially derived again from the crystalline part of the cellulose fiber. So this is not even is not a cellulose derivatives where some chemical process has been performed in order to change this group, the OH group of the native cellulose, but here we use only this crystalline particle itself, the colloidal particle. And actually what is really beautiful with this system is that the material becomes, it's solid, so you can maintain this helicoidal architecture in the solid film that they are able to form, and then you can make colored film, again, made only out of this pure native cellulose. And also here we really try to push the technology forward and trying to make material on large scale. And this is just an example that of really large film where we are able to control the interaction of the cellulose from the nanoscale to really to the meter scale to produce laminates that then we can cut and produce pigment out of it. So just to conclude, I think it's, we are really excited because we are able to really nicely control the preparation of the cellulose, and we are able to make material using manufacturing technique that are scalable, like this roll-to-roll, but we also use printing. And this functional material that we make now, what we are trying to close this diagram in our vision is to really want to assess what is their not only economical impact, but the environmental impact, that when we release them on the environment, what is their role if they can be really degraded. And at the same time, we want to develop new methodology to how we can extract more efficiently this cellulose, mainly from building blocks that are waste, so from essentially food waste, but also used goods like used textile. So, and with this, I would like to conclude the talk and pass the word to Thomas, who will go a little bit more in detail in the work of the cellulose nanocrystal.
Thank you to Sylvia for that very kind introduction. It was funny that on her slide it says Dr. Thomas Parton, but on mine it doesn't. In fact, I just finished my PhD a month or two ago, so the title is still a little bit strange to me. And actually, I would like to talk to you this evening about one of the main projects that I did in my PhD with Sylvia, about understanding the self-assembly of these cellulose nanocrystals that Sylvia mentioned, and how we can use them to create what we call photonic pigments. I don't need to dwell for too long on the motivation and the introduction here, because you've just seen Sylvia's very nice talk about this. But essentially, we want to take cellulose from a variety of different sources and use this to create what we call photonic pigments, which is just a fancy name for these structures which produce color from light interference rather than by absorption. And in order to do this, I just want to recap something about the structure of cellulose in these biological materials. Between the macroscopic scale, what we see of the cotton flower or wood or any other plant, and the molecular level, the structure of the individual polysaccharide of glucose, there is a whole hierarchy of different levels of structure. And in particular, if we look at these cells under the microscope, electron microscopy or optical microscopy, we see that the cell wall contains a whole mesh of different cellulose fibers. And as Sylvia has already mentioned, these cellulose fibers can be shown to be made up of bundles of smaller microfibrils, which themselves are made of individual cellulose chains. And so the structure here is really hierarchical. It's like when you make a rope, you start with a single thread and you weave this into a yarn and into a larger and larger structure. And the perhaps slightly surprising thing here is that if we look more carefully at the cross-section of these microfibrils, we find that the cellulose actually forms a crystalline structure. So we actually have individual chains of D-glucose, the individual cellulose chains, stacked together into a regular configuration. And actually, this is really essential for the mechanical strength of cellulose, which is why plants like to use this as a material, but it also leads to interesting optical properties such as birefringence, which we can exploit in order to create the colorful effects that we want to achieve. And so how do we make these colorful structures? Well, we start from natural cellulose fibers, and then using acid hydrolysis, we essentially break down the bonds along the chain axis for the cellulose, and we can create these cellulose nanocrystals, or what I'll refer to as CNCs, which are essentially pieces or segments of these microfibrils that we cut out of the natural cellulose fibers. And so you can see at the bottom here in this electron microscopy image that they're a couple of 100 nanometers long. They're smaller than we can see by eye, and they're more or less rod-like in shape. Now, what we find is that these are stable in a colloidal suspension. And when we increase the concentration of this suspension and really pack the CNCs together, they spontaneously assemble themselves into this helicoidal type structure, which was also introduced very nicely by Sylvia. So locally, the CNCs are aligned with each other, but the overall structure has a left-handed twist. It has a helix-like configuration. And actually, when we look at this liquid suspension under the optical microscope, we see this fingerprint-like pattern due to the periodicity, due to the sort of regular patterning of the structure. And so then if we take this liquid suspension and dry it out, we can create a solid structure which preserves this periodic arrangement of the CNCs. And what we find is that due to constructive interference at some wavelengths of light, some colors of light and not others, it appears blue in reflection because the blue light is reflected while the other wavelengths of light are transmitted. So actually, this phenomenon has been studied for a couple of decades now, but there's a few things that we still don't really understand about what's going on in this process. And something that's really bugged me throughout my PhD is essentially why the CNCs always choose to form this left-handed structure. They only ever form a left-handed helicoidal structure, not right-handed, and it's not immediately obvious where this breaking of symmetry, where this chirality or this choice of handedness is coming from. Because there are a couple of theories which have been proposed over the decades to explain what might be happening here. So one idea is that the individual particles are somewhat twisted or chiral, as we say, in the shape, a bit like screws or fusilli pasta, and that essentially, due to hard body interactions, they prefer to pack together in a twisted fashion. Another idea is simply that the distribution of charges on each individual CNC is itself helical, and that this would lead to a twisted interaction. The problem with all of these theories in the case of CNCs is that when we actually look more carefully at what the particles really look like, we see that it's actually quite complex, which is a polite way of saying it's a bit of a mess. We don't synthesize these particles from scratch. We extract them from a natural source. And as a result, they're highly varied in their size and shape. When we look more carefully, though, we see that the individual particles actually seem to have a composite structure. They appear to be made up of some kind of fundamental units laterally connected together into a structure which is maybe a bit like a Kit Kat chocolate bar with individual fingers laterally bound together. And I mean, the fact that it has a composite structure makes sense when we understand what is happening with the biological microfibrils that I mentioned earlier. So it seems that the idea that I had in my PhD was that perhaps these shapes, this morphology of the CNCs was important for understanding how they assemble into this left-handed structure that we use to make these photonic pigments. And there's another piece of experimental evidence that seems to really suggest that the shape of the CNCs is important. And that's the fact that we can start from quite bundly, quite composite CNC particles. And if we apply ultrasound, so like very high frequency sound at very high intensity, we can break apart these CNCs into the smaller subunits that they're made up from. And so I should emphasize here that this is ultrasound in the sense of like ultrasound imaging that is used in like medical applications, but the frequency range and the intensity range is completely different. So when you do a scan of a pregnant woman, there's not a risk that you're going to break things apart in that case. So here we apply very high intensity ultrasound. We break apart the CNCs, essentially tear them apart mechanically into smaller pieces. And what we find, interestingly, is that when we then use that to make one of these colorful films, we get a red shift in the color of the final film. So as we increase the amount of ultrasonication, we go from a blue film to a green to a red, as you can see here in the microscopy images. And essentially what's happening is that this helicoidal structure has a larger periodicity, which means it's twisting less. And as a consequence, it reflects a longer wavelength of light, and that causes the apparent color to go from blue to green to red. And eventually it will reflect in the infrared and we won't see any visible coloration at all. So this suggests that when you apply this ultrasound and change the shape and size of the CNCs, you change how strongly they interact in this chiral, in this twisted fashion. And so to basically summarize the last couple of years of my PhD in a slide or so, what I did is I looked at the shape of the CNCs in electron microscopy images, and I traced the outlines of the particles in order to completely capture their shape. And the point is, by doing this, I was able to access a very wide range of morphological or geometrical parameters, such as their length and width, but also many, many others, which really gave us a lot of insight into the shapes of the CNCs and how they were being changed by this ultrasonication. And as the result of this analysis, we were able to classify the CNCs into 4 different populations. So the reason that the sample is such a mess, such a mixture, is that it has different CNCs at different stages of breakdown. And so we called these subpopulations aggregates, meaning really large irregular structures, bundles where there's just a few subunits connected together, a bit like the KitKat particle that I showed earlier, crystallites, which are really the individual subunits of crystalline cellulose. And we also found a population called distorted crystallites, where it appears that the crystal has been kinked or broken along the chain axis. And you can see that we chose these names so that they would neatly spell out A, B, C, D. And so by looking at electron microscopy images for a wide range of samples, I was able to quantify the relative amount of each type of particle, A, B, C, D, as we increased the ultrasonication dose. And what we see is that there is quite a natural progression from A to B to C to D. Initially, when we have no sonication, our sample is mostly aggregates. But as we apply the ultrasound, we break the aggregates into bundles. Over time, we break the bundles into crystallites. And if we go too far, if we sonicate our sample to death, we're kinking and distorting the crystallites. And at the bottom there, you can see in the little octagons, these are the microscopy images of the corresponding samples. And so you see that the color really does go from really sort of violet blue to green to red, and then gradually loses color entirely. And so what we noticed is that the color of the films and the strength of this twist in the suspension was strongly correlated with the relative amount of this bundle B subpopulation. In particular, here in this plot, you can see a property called the chiral strength, which is just a measure of how strongly the interactions in the suspension cause the particles to twist. And we see that this chiral strength is plotted against the relative amount of the bundle fraction. And you see that they're very clearly correlated with each other. Now, of course, correlation is not causation. It doesn't necessarily prove that these bundles are the source, but it is clear that there is a connection between the composite form of the CNCs and their assembly. And so our particular working hypothesis here for future work is that these bundles effectively act as what are known as chiral dopants. So this is a technical term that's used for liquid crystals, where in, for example, liquid crystal displays. So in a standard liquid crystal, you have an elongated molecule which forms a nematic liquid crystal, which is an aligned phase without any twist. And then you can add a certain chiral molecule, a molecule with either like sort of D or L enantiomer, and depending on which one you add, you can make the whole nematic phase twist in a left-handed or right-handed fashion. And essentially our idea here is that these bundles are acting as these agents which cause the other CNCs to twist. And so the reason that the ultrasonication weakens the twisting interaction in the suspension is essentially that the bundles are being broken apart, they're being blasted into crystallites, and so we're effectively removing the chiral dopants from the suspension. And so in terms of their 3D shape, because I've been showing you a lot of images here, but this has all been 2D. In 3D, we have evidence that the bundles have a morphology a bit like this sort of bow tie or propeller-like shape at the bottom there, where in fact I called them a Kit Kat before, but perhaps it's more of a, in 3D, it's more of a propeller-like structure where there's a twist along the direction that they're laterally connected. And so, yes, I've talked about pasta and Kit Kat, so perhaps I'm a bit hungry, and it's time to end the talk. I hope that you found this interesting, and I'd just like to finish by thanking the other members of the Vinolini group and the sort of funding bodies for supporting this research. So thank you.
So thank you, Sylvia. Thank you, Thomas, both for those contributions. And we now can move to Q&A. So I will start out by reflecting a comment from one of our participants who says that she's an artist and interested in building a database of primary colors. I suspect she's going to be contacting you. But I suppose I might just spin this into a question to you, Sylvia. Could you imagine, as it were, being able to produce a color, an artist's color box using this kind of approach?
Actually, there are lots of work into this context already, especially regarding the natural world, because this structural color have been observed in so many elements, and then people really try to characterize them and put them together, associate them to the structure and to the optical appearance that you have. And the more difficult, that this is also what maybe Rachel is intending to, is like to try to plot them within a context of what is the color vision. So I've never seen much done in the context of color vision for humans. Yes, so like really representing and relating it to primary colors and normal, the usual visual color, RGB or whatever other system you want to represent. But it's been done a lot in the context of animal vision, where the color spectrum is much more enlarged. And a lot has been done for birds, that they have more photoreceptors. And what they observe, and I think that's what I wanted to get to, that is really beautiful, is that this palette of this so-called photonic coloration of structural color increased the diagram in terms of occupied area of 40% with respect to the normal coloration. So the area, if you really map colors in a color diagram vision, like you can expand the richness of this diagram with the structural color really greatly. So I think this is also another indication that in nature are really important for communication. Yeah.
Okay, thank you. Now I think this question is for you, Thomas, and it's a more technical question, and that is Natalie asked whether you've considered using scattering techniques to characterize the distribution of the structures that you have. And she suggests this might be easier to do than looking using microscopic techniques. Have I made myself clear?
Yes, that's clear, and I think it's a very nice question. So, in fact, I didn't talk about this, but we have done some characterization of these particles using dynamic light scattering and static light scattering techniques. So we have done a bit of this. The nice thing about scattering is that you measure a very large number of particles, a truly colloidal ensemble, like billions upon billions of particles, but often you get quantities which are averaged over all of the particles. So in the sense, the values are very robust, but it's very hard in cases like this to get accurate idea of the morphology. I mean, even if you have perfect cylinders, but there's some dispersity, some variation in their length and diameter, the scattering curves very quickly lose any sort of fine features. So yeah, there is definitely a lot we can learn from scattering, and it's something that we've done. We also did an X-ray diffraction to have a look at the crystal structure. But so yes, this is it's also a valuable approach, which we also looked into.
Thank you. And I think just a slight follow-up on that, Thomas, if you just keep on the line. If you go to extreme sonication, do you have evidence that the samples will reflect outside the visible region? I mean, is that expected, or have you observed that?
Yes, so some of the samples that I showed, I just sort of put an X in the microscope image. There is actually a microscope image there. It's just that by eye it's transparent, but looking at the reflection spectrum, I can see that it's reflecting in the near IR. And we can push further than that. On the other end, it's also possible to push a little bit. You know, if you have no ultrasonication at all, it's also possible to push a little bit into the near UV. So we can do the visible range and a little bit beyond it. Yeah.
Okay, thank you. So I think this one is for you, Sylvia. The comment in here is that when you look at an insect, for example, the color varies with the angle that you look at it. And the question is that, will that be the same for the collagen nanocrystals? Is that a property that one would expect as well?
Yeah, this is, it's not really collagen. In this case, it's not collagen. Collagen is the case of some mammals use collagen. We use mainly cellulose. And this cellulose nanocrystal at the end, you can produce this color that change in function of the angle. But we have also developed a different system, adding some higher level of hierarchy. So where you can actually engineer the response that is angular independent. So in contrast with the pigment, where you can only have a color that depends on the chemistry, so you cannot really change how it changes in function of the direction that you are looking at. With structural color, the majority are angular dependent, and this is lots you find in nature, but also it's also possible to use them to make angular independent color. And we can do both, and actually in nature also you find both. So it's possible.
So I think just sort of synthesizing 2 or 3 different questions together for you, I think, Sylvia. So people are, of course, ask themselves the question, can you turn this into something that one could use practically? And how far are you along that? And somewhat related to that is the question of how robust or durable might you expect these materials to be? So thank you for this question.
Yeah, I'm really happy to answer, because I'm really proud that one of my PhD students, Benjamin, essentially launched recently a company that he called the SparkCell for sparkling and cellulose. And his really aim is to try to really scale up the production of this colored pigment. And regarding in terms of stability and characteristic that you can get, it depends on what you want. So this pigment itself, they are made of cellulose. So if we don't do any treatment, they can be, you put them in water and then they will simply redissolve. Yes, if you apply a bit of shaking, they will redissolve. But if you apply a treatment that is also a really simple thermal treatment without adding any chemical, the material becomes stable for years, because somehow the cellulose is kind of fusing together, and then the water is not able anymore to permeate the structure, and actually it almost doesn't swell it even anymore. So with really simple thermal treatment, you can make them much more stable. So it depends on the final use. And obviously what I think it's important to remind, and I think it's important to think about, especially when you talk about this biomaterial for application, when you want to use them in application, this is one of the many bits and pieces that you use to make a color. So the pigment is one of the ingredients, but you have lots of many other material that you need in a formulation. And in terms of stability and different characteristic, resistance to water, to environment, other chemical environment, it really depends also on the other ingredient of the formulation. But I saw the question, they say like, would it resist to a hot wash? The answer is yes, because cellulose is thermally stable up to 250 degrees. So this is, I think it's one of the really extremely good properties, just because it's highly crystalline. So this would be true for the nanocrystal, not for the cellulose derivatives. Yeah.
Right. Okay. Perhaps Thomas, you could answer this one. So this is a rather specific one, but it's quite fascinating. Maybe, and the answer might be, I don't know. But our participant says that his wife has a titanium ring, which had been colored by the jeweler passing a current through it. And then the color depends on the current that's passed. And he wonders whether you can explain what is going on there. Is this related to what you're doing? If you're looking worried, Sylvia's going to answer.
Yeah, I see that Sylvia is nodding, so I think perhaps I'll give it over to Sylvia, because personally I'm not totally clear, and I think Sylvia has an idea.
Okay, Sylvia, sorry.
So it's a technique that you can actually use to create porosity on metal, on also different other material. And this is one of the first technique actually invented to make a photonic structure, or really simple, what you call Bragg mirror, for application also in optics, for making interference filter and things like that. So it's still the same phenomenon of structural color. You make a layer in the material that has a different porosity, so different refractive index. So, and because also probably it's a metal, you have a lot of titanium, so you have a really strong refractive index contrast between this different layer. And then you can, if the layer they are on the right length scale, so they are the right size, you can again interact with light with this phenomenon that is interference, and you make color out of it. So this principle of structural color is essentially really simple. It's really powerful. You just need to, as long as you are able to control material at the scale of the wavelength of the light, then you can really design the optical appearance that you want independently from the material that you use.
Okay, good. I knew you know the answer. So just going back to you, I think, Thomas, is the question I had in my mind, and it's also reflected in a couple of others that are in the Q&A, and that is about the process of going from the crude cellulose to the material that you want. You just said, oh, acid digestion. But I mean, presumably what you get depends very, very much on the processing, or does it not?
Yes, you're quite right. Yes, so I glossed over this, but what we do is a sulfuric, an acid hydrolysis using sulfuric acid. In fact, actually, one of the challenges that we have at the moment is that this process is it involves using quite highly concentrated sulfuric acid, something like 50% by volume. It's really, and this really makes sense because as anyone who has eaten fiber or roughage will know, you can consume cellulose and it passes through the acid in your stomach and out of your digestive system, and a lot of it comes back out the other side unaffected. So you do need to use quite aggressive conditions in order to break apart this crystalline cellulose. And the point is, we don't want to completely dissolve it. We just want to fragment it. And there are greener ways of doing this as well, using, for example, enzymes. And like some people in our group are working on improving this process. But essentially, yes, we're in this study, I used sulfuric acid. And yes, by tuning the temperature and the concentration of acid and the duration, we can tune to a certain extent the length and the cross-sectional area of the particles that we produce.
But I see what you're saying is, in summary, is in the current form, this is not a process that one should try at home.
No, certainly not. No.
Okay, but there is hope of finding perhaps a more gentle way of doing it.
Yes, yeah, there seems to be very promising ways of doing this. Of course, cellulose is truly completely biodegradable. There are, ways of doing this. It's just that we haven't figured out a way to do this in a, at a large scale, in a sort of commercially viable manner.
Yeah. So I think the next question here, again for you, Sylvia, is perhaps taking this more broadly about, are there other ways that you can see on the horizon of making biodegradable colored materials? I mean, are there other things to explore here?
Yes, we have investigated, I think we prefer the cellulose, but we have investigated other polysaccharides, for example, for chitin. and also starch particles. So it's a huge variety of material that in principle you can use, because the difficulties is only really controlling the assembly and controlling the size where the structure are made. We also work a lot with the synthetic system, but biodegradable polymer, for example, that we have demonstrated that you can also exploit for the production of this material. And then many other groups are investigating other bioproducts. People are working with collagen, guanine, and depending on the system that you are particularly interested, also in nature, you have the counterpart of the material science that there are examples. We believe in cellulose because we think it's in principle can be extracted from many waste, also bio waste, including food. And that's why we mainly focus on that. But in principle, there are many other biomaterials that can be used for the same approach.
Thank you. And then I think this is a tricky one, Thomas, but I'll try. So you explained why you think the CNCs formed helices. Is there anything to say about why they are left-handed rather than right-handed?
Yes, this is the million-pound question, really, and the short answer is we think that it ultimately derives from the fact that the cellulose is made from D-glucose rather than L-glucose. We think that fundamentally it arises from the molecular chirality of the glucose monomers, of the glucose monosaccharide. However, the question is not so much like where does it come from, but how does it get transferred across length scales from the molecular level across the, up this hierarchy that I mentioned earlier, up to the level of these bundled CNC particles. And that's actually something that we're not entirely clear about at the moment. It's actually the topic of my current doctoral fellowship in the Department of Chemistry. But essentially, we think that the individual crystallites have a mild twist along their axis, and that then this comes directly from the molecular chirality of the glucose and the cellulose, then this mild twist gets turned into this much larger twist when they pack closely together into these bundles, which happens in the natural tissues. And we also have evidence to a certain extent that we can control this sort of directed aggregation to create these sort of propeller-like particles. And then this determines the overall handedness of the large structure. And actually it's part of a sort of more general phenomenon which we see in nature, and we also see in synthetic systems of chirality being transferred across length scales. And so it's really interesting as a fundamental phenomenon. I think that's why I'm sort of really fascinated by this, but it's also useful obviously for creating these colorful materials.
Thank you. I think that brings us to a very natural conclusion here. So I'd like to thank you, Silvia and Thomas, for your talks. Thank you very much for everyone that's dialed in. I hope you found it interesting. There will be more of these, and we will inform you about them in the usual channels. And in fact, you should find that the latest printed edition of Chem at CAM, if you subscribe to that, has either landed on your doormat over the last few days or will do so soon. So thank you very much to everyone, and good evening.